Hello everybody, I want to spend today talking about mitosis. So mitosis is normal cell division. Today we're going to go over the phases of mitosis and sort of what it's for in both single-celled and multicellular organisms. If you have that worksheet that we've been doing in class, you're going to want to fill this out as we go through it. Most of us did this in class, but if you missed class, this goes over it. So remember, the cell cycle, which we went over last week, consists of the entire life of a cell, if you will. Most of the time, cells aren't dividing. They're doing other stuff. They're making proteins and potentially going through the process of getting ready for division, but they're not actually dividing. Here's the entire cell cycle, starting with the end of mitosis. Here you notice we have a cell that's got 2N information. That's a normal cell. And as we play through it, this cell with a normal amount of information does protein synthesis, builds organelles, does whatever it's supposed to do for that part of the organism that it's in. And if it reaches this checkpoint, and if it has enough juice, if it has enough energy, has built enough parts, it'll start that process of preparing for mitosis. If it doesn't, if it reaches this checkpoint and it doesn't have enough energy, it'll enter the G0 or resting phase. Otherwise, it goes from G1 and starts going into the S phase. And in the S phase, DNA replication happens, resulting in it going from 2N to 4N information inside its cells. After that, it goes through the G2 phase. Again, in the G2 phase, it's just doing protein synthesis, building up its more stuff inside the cells. At the end of the G2 phase, if it's ready, it'll enter the process of mitosis. And there's a checkpoint in mitosis, but essentially it goes through the process of division, where you go from one cell with 4N to two cells with 2N. So, with that, let's spend our time now talking about the process of mitosis itself. So mitosis is essentially normal cell division. It starts with one cell. The end result is two cells that are almost identical to that original cell. They're definitely identical in terms of the information they have. You start with that one cell at the end of that G2 phase, where it's got 4N information, splits out to two cells with 2N. But that's enough to rebuild to 4 and go through the same process again. So in order to talk about mitosis, we need to begin by identifying the actors or the parts involved. So this organelle here, the thing that separates the cell from the outside world, is called the cell membrane. It separates the cell from the rest of the world. It's a membrane and it's made of lipids, and it separates that cell. The stuff that makes up the inside of the cell is called cytoplasm. And so cytoplasm is the jelly-like substance that makes up the internal parts of the cell. Very water-based, but there's a little bit more to it. This thing, of course, this large structure inside the cell, is the nucleus, and it's found in all eukaryotic cells. And so we can go over that in some more detail later, but the nucleus is a really important part of eukaryotic cells. And of course, what's stored inside the nucleus is DNA. And in this form here, the way we see it in this picture, we call it chromatin. It is DNA, it is chromosomes, it's just kind of a jumbled up mess of them, and so we refer to it as chromatin. The part that separates the nucleus from the rest of the cell is, like the cell membrane, separating the cell from the outside world, but in this case, it's the nuclear membrane that separates the nucleus from the rest of the cell. For these little organelles over here, the centers of those bodies are called centrioles. And these centrioles are really important in cell division. These little legs that come off of the edges of them that look like spider legs, these are called spindle fibers. And these are all going to play a role in mitosis. The first phase of mitosis that we're going to talk about is interphase. And technically, interphase is not a phase of mitosis. It's everything else that's happening. It's the G1, the G0, the S, the G2 phase. It's where everything else is going on inside the cell. So it technically isn't part of mitosis, but it certainly is really important. It's when everything else is going on for the cell. When you look at an actual cell under a microscope, what you'll see for interphase, the really good distinguishing characteristic, and this is certainly worth writing down, is that nucleus, you can't see individual chromosomes. It's sort of this dotted out black space that maybe it looks a little brown or looks a little bit of it, that maybe has a little bit of color to it, but for the most part, you can't see individual chromosomes. That's how you know you're in interphase. After interphase, we have prophase, and this is the actual beginning of mitosis. And in prophase, there are three things that really distinguish it from interphase. Number one, the spindle fibers stretch out across the entire cell, so we see them stretching from one side to the other. Number two, the nuclear membrane, you see, starts to break down, and the nucleus starts to fall apart a little bit. And number three, and probably most importantly, we can now see individual chromosomes. So if you're looking at a cell under a scope, what you'll see here, you'll still see the same nucleus, but you start to see individual chromosomes. You see those darkened lines present in the nucleus for the first time. It's not going to look perfectly clean like it is in the model here, but you are going to see those individual chromosomes. So that's prophase. After prophase, we come to metaphase. And in metaphase, the nucleus is completely gone, and all of the chromosomes are lined up in the middle of the cell. You'll also notice that the spindle fibers attach from the centrioles to what's called the centromeres, which are the things that join the two chromosomes together. So the chromosomes are lined up in the middle, and they're attached to the spindle fibers. Again, what would you see in a real cell? In metaphase, again, you typically don't see those spindle fibers, but what you would see is a whole bunch of dark coloring of those chromosomes right in the middle of the cell. So you do see something in the middle. After metaphase, we have anaphase. And in anaphase, we start to see those chromosomes being pulled to opposite sides of the cell. It's like a fisherman has hooked those chromosomes and is pulling them in. And since he's pulling in from the middle kind of goes first, and you see these trailing ends. So what you would see under an actual cell is these sort of V-type structures moving back towards the ends of each side of the cell. So now we're pulling those chromosomes apart, getting them ready to split, so they're leaving the middle and they're getting pulled off to opposite sides. After anaphase, we have telophase. And in telophase, we start to see the cell getting split into two, and we start to see two nuclei forming in these, what are going to now be the two new cells. So it starts to split down the middle, and we start to see the formation of two new nuclei, one in each of the new cells. And finally, the process is ended with what's called cytokinesis. Cytokinesis, cyto is cytoplasm, kinesis is cut. So in cytokinesis, we see the cutting of the cytoplasm, and now we have two cells in interphase, ready to start the G1 phase and go through their own process of growth and development before they divide on their own. When you have a process like mitosis, sometimes it's helpful to have several mnemonic devices to help you remember them. Here are the ones that I use. You may have your own, but here's what I use to remember the stages of mitosis. First of all, IPMAT here helps me remember the order of the phases of mitosis. That might help you. It might be worth writing down somewhere. But now I'm going to go through each individual phase and what I use to remember it. So to begin with, interphase. Interphase is what happens between mitosis events, and it's not very interesting from a division standpoint. Sure, a lot's going on, G1, S, G2, and so on, but interphase doesn't look as interesting as these other phases. It's also, if you're a theater person, interphase is like intermission. It's the part in between the scenes where nothing is going on in terms of the play. So interphase, not very interesting, or intermission. In prophase, pro means before, and so that can help you. It's the very first phase of mitosis here. I remember it as a pro quarterback, like Touchdown Tommy here, the GOAT, throwing his touchdowns to get his 7th Super Bowl ring. To me, the spindle fibers here look like a quarterback throwing a ball from maybe from Tommy to Gronkowski or whatever. But that works for me. So prophase is like a pro quarterback throwing those spindle fibers out across. Metaphase for me, easy to remember. In metaphase, I use this MMM system. Metaphase, they meet in the middle. In this phase, all of the chromosomes line up in the middle, ready for division, and it all starts with M. So meta, meet, middle. That's what helps me remember that. And forgive me for my kind of ridiculous ones for the last few. In anaphase, I think of Little Orphan Annie, which was a show I was obsessed with, or a play I was obsessed with when I was a kid. Annie is an orphan who gets adopted by this rich guy, Daddy Warbucks, and at some part in the film, or in the play, she gets taken away from him, or ripped away from Daddy Warbucks. Thankfully, in the end, she gets reunited, but that part of her getting ripped apart is what I associate with anaphase. In this phase, those chromosomes are getting ripped apart, so anaphase, little orphan Annie, just like Annie gets ripped away from Daddy Warbucks. And after anaphase, we have telophase. And in telophase, the two chromosomes are separated now and need to talk on the telephone, if you will. And so since they're often two different nuclei, they have to call each other on the telephone. That's how I remember telophase. And then finally, for you South Park fans, for me, for cytokinesis, every single episode of the classic cartoon South Park, Kenny dies, and they say, oh my God, they killed Kenny. So you can imagine getting cut in half. That would certainly kill you. And so cutting the cytoplasm in half would kill you. So, oh my God, they killed Kenny. Cytokinesis killed Kenny. Again, it works for me. It might not work for you, but that helps me remember what happens here. It literally means cutting the cytoplasm. So maybe that works for you too. If I were to cut off some of my skin cells and put them under a microscope, what you might see is something that looks a little bit like this, where there are a whole bunch of cells in interphase. They're in the process of just going about their normal lives. Now, if I happen to find an area where there's a lot of growth going on, like the tip of a root or an agave plant that's growing its stalk, or maybe in one of our baby axolotls that's developing a lot, I might see something a little more like this, where most of the cells are in interphase, but a few of them are in various stages of mitosis. So prophase or metaphase or whatever. And this is what you normally would see in an area with a lot of growth. And in a lot of classes, you'll actually get a chance to do this, where you take a look at a slide of an onion root. And these onion roots are growing a whole bunch before they cut them and dyed them. And if you zoom in on a section of an onion root, you can actually see individual cells in different stages of mitosis. So here, take a minute to identify A, B, C, and D. You can pause this video for a second, but then I'll go ahead and answer those up. Okay, hopefully you paused it and didn't just go straight to the answer. A here is, of course, anaphase. Anaphase is when we're starting to see those cells, those chromosomes getting split to opposite sides. So A would represent anaphase. I think not only is A anaphase, I think there's a lot of anaphase cells out here. So here's anaphase, here's anaphase. That might be really late anaphase over there, whereas you have really early anaphase here. Another anaphase. Lots of, there's another one maybe. That might be late in it. But so those ones are all anaphase. B to me looks like telophase. Telophase, you might be able to argue this one here is also telophase, but much earlier, very end of anaphase. This one here may be telophase. You can talk about this one being telophase, if not, just after cytokinesis to an interphase. Same here, very late telophase, starting to go into two new cells. Really late telophase, I would say that's just after cytokinesis here. For C, C is a tough one. I think you might be tempted to say C is interphase, but it's not. It's prophase. And again, you can see those individual chromosomes. And in prophase, to contrast it from these cells next to it, most of these other cells are in interphase. But in prophase, we start to see individual chromosomes. So prophase, we're seeing individual chromosomes. Prophase, prophase down here. Again, you're starting to see individual chromosomes. That's an indication that it's prophase. And finally, D is metaphase. There's some others in metaphase here. That might be early metaphase. Here again, all lined up in the middle, other examples of metaphase. All right, so hopefully you understand all the different phases of mitosis and what we would see. So hopefully you got all that and can identify these different phases. There are a couple other questions like, what is mitosis for? If you're a single-celled organism, mitosis is how you reproduce. It's how you make a copy of yourself. So if you're a protist, like a euglena or a paramecium or an amoeba, this is a very similar process to how you would make a copy of yourself and reproduce. For us humans, this isn't how we reproduce. We reproduce sexually. So it's a little bit different for us. It's not like when I had my son a few weeks ago, I just got a copy of myself that came off. It was a little bit of a different process, which we'll get to next week. It involves a different type of cell division. But for us, mitosis is how we grow and how we repair some and replace some of our cells. So my son, being a very young kid, is going through a lot of mitosis right now as he continues to increase his number of cells. The same is true for all of our axolotls. They're doing mitosis to grow and develop. Hopefully that helps. If you have questions, feel free to message me.